Hello, hi everyone. Can you all hear me? I feel it's quite strange to use microphone. So if you are okay, I will just present this way. Yeah. So good afternoon. And first of all, I want to thank the organizer to give me this opportunity to share the work from my group today. And my group is quite new, so 2 years ago, I moved to Singapore and started my own group in the Genome Institute of Singapore. And then my personal background is in gene regulation, epigenetics. But now we are more interested in the signal from the body, especially the dietary factor. That's why we start doing research focused on gut microbiome, and we also developed the tool. Hopefully it can help the whole community to gain the new insight on the molecular level. Yeah, because from the talk this morning, we see the lipid or the oil, actually, they are very important for the health. And now I think the next step, we want to learn more about what's the underlying molecular mechanism. And then that's the research direction we are working on right now. So first, I just want to give you a very brief summary to show you how complex it is between the diet, host, and microbiome. Yeah, so as we all just finished a nice lunch, so you can think about, after we eat the food, it also the nutrition, the dietary component, once it enters the body, now people think the food is medicine. So how our body responds to those dietary factors is important. So the host genetics will play an important role here. And then once it go into the gut, those dietary components will start to interact with the bacteria living in the gut, and there are studies showing they can shape the composition of gut microbiome, and that will lead into change the metabolite secret from microbiome community, and then it will become a feedback going back to the host, and then give different phenotypical change in the host. So in today's talk, I'm going to share with you the study we are looking at. We try to understand what's the effect of the microbiome. And then here we are focused on cardiovascular disease. Yeah, because it's an important disease, and then it's also affected by the diet. And the second part of my talk here is, we want to focus more on host genetics. Because now we know it's become more affordable to sequencing almost everything. Yeah, so for example, human genome, microbiome, the bacteria, we can generate a lot of DNA information. But actually, how can we apply those information in this complex interaction? I mean, because we know the host genetics will play an important role for the personalized response. So consistent with that, recently in US, they also started a large project called All of Us. I think they are doing a cohort study. They try to collect a lot of genetic, microbiome, dietary information. And lastly, here is something I'm looking forward to learn from this composition. Today, from the conference today, we know the dietary component is play a huge role and impact on the host phenotype, and also it will shape microbiome composition. So I'm looking forward to the discussion with all of you during the conference. So I can, it's also a learning opportunity for me. Okay, so let's start. So we know the DNA sequence in our body is fixed, but actually our genome is a response to the environmental signal all the time. So that including diet or air pollution or just the lifestyle in general. But when we look at our genome, only 2% of them belong to protein coding gene. So that's usually the part people trying to generate, for example, RNA-seq, or they try to profile the pathway, the metabolism, metabolite pathway. But actually, there is a huge part of our genome, 98% of them belong to non-coding region. And from almost 2 decades study leading by the ENCO. So ENCO is a consortium looking at what's the function of those non-coding regions. So now we start to have an idea, actually, those non-coding regions play an important role to control gene expression. So basically, it can sense the signal from the environment and tell the different cell type, different tissue, how to respond to those signals. So here I just draw a very simple cartoon to explain. So for example, in this study, when people study cancer cell, and then here they are looking at MYC oncogene. So we know MYC is an important oncogene, is upregulated in many cancer types. And here they are doing the image approach. So they are looking at the enhancer control MYC. They label in blue here, and then they also have another probe label the MYC promoter in yellow. So here they can see when they turn on the notch signaling pathway, they can see those 2 dots will come together and become white here. But once they shut down the notch signaling pathway by chemical inhibitor, they can start to see those 2 pieces of DNA start to separate from each other. So yeah, basically that tells us in the new case, in the chromatin, the organization is continually dynamically change based on the signal from the environment, and that will change the gene expression in the cell. So the current model is, we know the gene expression is controlled by promoter and also controlled by the distal regulatory element. And when the external signal comes in, it will trigger more interaction to either, for example, activate the gene expression here, and then that will cause, for example, maybe promote cancer cell proliferation or activate immune response. And this gene regulatory mechanism is fundamental in all the cell type. So that's the topic we are studying right now. So for example, the key question right now is where are those functional regulatory elements in vivo? Because a lot of study has been done recently. They use cell line or organoid model. And we know it's an artificial condition, right? You don't know what's the impact of a dietary component. So that's why I will share with you recently we set up a system to identify those elements in vivo. And why is that important? I think that's the question I got asked a lot. So from the previous cohort study, people already show by genome-wide association study, you can start to correlate certain risk variant associated with different disease. So now they encourage people, maybe the genome sequencing can be a great tool, right, to tell you once you carry this risk variant, you have higher chance to develop certain disease, but what can you do next? So that's why I think it become the limitation. A lot of people, they become hesitate to do the genetic test because once they know the genetic information, nothing will change. But actually from this paper published in New England Journal of Medicine about 6 years ago, they tell you genetic variant or risk is not the only factor. So here, when they look at the cardiovascular disease, they found for the people who carry high-risk genetic variant, if they have favorable lifestyle, so which means if they eat more healthy, do more exercise, actually the risk for them to develop cardiovascular disease actually will be quite lower compared to another group of individual who carry the same risk variant, but they don't have the good lifestyle. For example, if they continue eating junk food or continue doing what they are doing. So that creates an opportunity. Actually, the genetic sequence is not the only factor to determine the disease progression. So in our first study, we want to know, so the lifestyle here cannot be gut microbiome. Because we know a lot of lifestyle eventually will go to the gut, and then the gut microbiome is like a key resource to sense those signals and then feedback the signal to our body. So the first study we carry out before I start my own group is we want to know what's the role of gut microbiome for atherosclerosis. So here we use the mouse model. Because we know dyslipidemia, this disease, there are a lot of genetic mutation associated with this disease. For example, one is the mutation in LDLR, the low density lipoprotein receptor. And then in this mouse model, if you keep the mouse on the regular diet, they are totally fine, they don't develop any disease. But once you put those animals on the Western diet or high-fat diet, after 10 weeks, you can start to see the lipid accumulate in the heart, and then you will develop atherosclerosis. And then when you sequencing the composition of gut microbiome from those animals, here you can see when the animal on the Western diet, the abundance of firmicutes labeled in green here will become more abundant, and then we also see the decrease in the bacteroides. So now we are asking a very naive question. I mean, because it's always difficult to ask people change diet, right? Because it's difficult to convince people to do that, even for smoking. So here we are thinking of what will happen if we can have a way to remodeling gut microbiome without change diet in the animal. Can we provide, if we can do that, I mean, first, can we do that? And second, what will happen if we can do that? Well, can we see any beneficial effect? Because the animals will be still on the high-fat diet. So that's why we decided to use this chemical molecule called self-assembling cycling peptide. So it's a short peptide, contains 6 to 8 amino acid group. And then here you can see we on purposely incorporate 2 different types of amino acid. We put in D-form and L-form amino acid. And the reason is once they interact with the membrane, they can base on the interaction between intermolecule interaction and form the nanotube structure on the membrane. So that will penetrate the target bacteria, and also this interaction is sequence dependent. So that means we can change the sequence here on the cycling peptide, and then we can provide the specificity to deplete or inhibit the growth of certain bacteria in the microbiome community. And the first thing we test here is we test a bunch of cycling peptide with different amino acid. And then here we use commensal gut bacteria. We perform the MIC assay. So here you can see, depending on the sequence, they have a different effect on different type of bacteria. And then another advantage of this peptide is we can synthesize them, so we can provide the proper control. For example, the candidate peptide we see here can inhibit some firmicutes and bacteroides. And then here, if we just put a methyl group on the side chain, we can also disrupt this impact on the gut bacteria. And then actually this cycling peptide is not brand new, built in the bacteria. As you already know, there is an antimicrobial peptide. So in the community, they already have the natural product to tell other bacteria, you should not come to my space. So I think we are inspired by that observation and develop this cycling peptide. Okay, so now it seems the cycling peptide also work on gut bacteria. The next thing, we set up a high throughput screening system. So here, we isolate the sequence from the animal, and then we optimize the condition to grow the microbiome community in the lab. So in one tube here, it contains more than 100 different species. And now, if we apply this culture into 96 well, and then each well we can put in different peptide sequence. And then after overnight incubation, by doing sequencing, we can identify which sequence affect targeting which species. And then here is the heat map here, just simply show you which species their abundance decrease after the treatment. And one thing more interestingly here, when we perform the clustering, actually here we can see 6 unique clusterings. And then which means the peptide or chemical we use in each cluster, they will targeting the same group of bacteria within the community. So for example, the ampicillin or chloramphenicol, the broad spectrum antibiotics, we see them are present in the same cluster. And then based on the data we generate here, we want to provide a ranking criteria. What's the good candidate peptide we want to use in our study? So here the hypothesis was, we assume there are certain, we call bacteria or some bacteria might contribute to the disease when we put the animal on the Western diet. So we will see their abundance will increase when the animal on the Western diet. And now we are looking for the peptide which can bring their abundance down or closer to the child mice and without kill all the community. Because if you just put a bleach into the culture here, basically you kill all the bacteria, but you don't learn too much here. So based on this ranking criteria, we come out to top candidate for the animal study. So in the animal study here, as I told you, so here, the animal still on the Western diet, but at the same time, we provide the cyclin peptide in the drinking water for 10 weeks. So every day, the mouse will continue eating, and then when they drink water, they also uptake those cyclin peptide. So here, we want to know what's the phenotype on the animal. So here we are checking gut microbiome composition, body weight, toxicity, or the phenotype change. So the finding here is after 2 weeks treatment, we can already see the separation between Western diet, which labeled in green here, versus the peptide group. So from the PCA plot, you can see that the composition of gut microbiome, they start to separate after 2 weeks treatment. And then what happened on the animal here is, after 10 weeks, when we sacrifice animal and do the lipid staining, we can see now we have more lipid staining on the heart here, and then it's more significant. And when we have the control here, it doesn't affect too much. And then we also notice the cholesterol level is significantly reduced after the peptide treatment, and this reduction is microbiome dependent, which means if we deplete the gut microbiome by antibiotics, and then after that we give them peptide treatment, and then we don't see any change in the cholesterol level. And then we also find the reduction is coming from LDL and not from the HDL. And then we also monitor the cholesterol absorption in the animal. Because one possibility is maybe after peptide treatment, the animal, they don't absorb the cholesterol from the diet more efficiently as they usually do. So here you can see when we measure the cholesterol level in the plasma or in the feces, actually we don't see too much change in the first few days. But after 2 weeks, we start to see the whole body, the cholesterol level, decrease. So that means the bacteria in the gut somehow, they can affect the cholesterol metabolism in the body. And then here we also identify several candidate species which are correlated with cholesterol level in the animal. And then here, you can see for those species positively correlate with cholesterol abundance after peptide treatment, we also notice their abundance becomes significantly reduced. And then another phenotype we also find consistent with a lot of previous study. We also see after peptide treatment, the immune response is reduced. And then we also find the T helper cell, their abundance increase, and the release of several cytokine, they also decrease. So it's all like it's like the bacteria is doing multiple things in the body. And then here we also look at the gene expression change in the liver. Yeah, because we know liver is the key organ to regulate metabolism in the body. So here by RNA-seq, we can see two groups of clusters are affected after the peptide treatment. And then the cluster 1 is related to lipid metabolism. And then here we see the gene from that cluster become upregulated after peptide treatment. And we also confirm one of the target gene, CYP7A1, the expression is become induced after the peptide treatment. But from here, it becomes very interesting. So how does the bacteria in our body can change gene regulation program in the host? And from the literature, we know a lot of metabolites such as short-chain fatty acid, people already show it can regulate epigenetic regulation in the host. But here we want to have a more systematic measurement. So here I just skip here. So one thing we did first is we want to know is any epigenetic change after peptide treatment. So here we perform ChIP-seq looking at histone H3 K27 acetylation, which is a histone mark associated with gene activation. And then here you can see by compare red and blue here. So red means the animal on the high-fat diet, and then blue, purple means on the high-fat diet plus peptide treatment. We start to see a lot of stronger histone H3 K27 acetylation signal increase after peptide treatment. But more interestingly, when we look at a T-seq signal, a T-seq can measure the chromatin accessibility, tell us in the genome which part is accessible. But when you look at the openness or accessibility of a chromatin, actually it doesn't change between the treatment. So that means the signal secreted from bacteria actually regulate the histone modification on the specific region in the genome. So what's the role of the gut microbiome in host gene regulation? So in my group recently, we set up the system to address this question because we think it will provide the underlying molecular mechanism. So first, we want to know which regulatory element is functional in vivo, and second, which regulatory element actually can sense the signal from gut microbiome. So here, just a very brief background introduction about the reporter assay. So people already developed or used reporter assay for a long time. So what they did in the past, usually once they identify a interest of a genomic region, they can clone that genomic region into a reporter construct, for example, luciferase or LEC, and then they can inject to the mouse embryo, and then here you can see from the whole embryo staining, they can start to see the reporter gene only show up in a certain part of the body. So they say this piece of DNA is important to regulate gene expression, for example, in liver in this case. But the problem is this assay is very useful, but it's a very low throughput. Very low throughput means you can only test one element at a time. So we know in our genome, in human genome, there is a 3 billion base pair. So recently, people developed another approach called MPRA, massively parallel reporter assay. So how it works is you can generate a library. So in the library, each construct will carry the DNA element you are interested in. And then here you can incorporate, at the same time, if you incorporate molecular barcode into the reporter gene, now when you introduce the whole library into cell or tissue, which we develop, I will share with you later, and then by measuring the barcode abundance, you are able to profile, generate the result altogether. Okay, and why is that important? Because people also show, when use this assay, they can start to look at, for example, if you can put in the same element, one contains, for example, reference sequence A, and another one contains risk variant C, and now you can see how those two elements are under the condition after treatment. So here, when you do the assay with or without drug treatment, they can start to see the change in the response. So here, yeah, we pick the liver as the organ, yeah, to do the study, because we know, yeah, liver sends a lot of signal from gut microbiome. And here, another thing we also improve here is we try to make the model more mimic to human condition. Yeah, because a lot of mouse study, they are looking at pathway signaling transduction in the mouse. And my colleague asked me, how is that related to human study? So here, we purposely look at the human DNA element identified from human liver. So they are previous work, they already profile and identify the candidate regulatory element from human liver. So overall, we construct a library contains about 110,000 elements. So we introduce the element into mouse liver, and we want to know what's the response of those elements in vivo. And then here we can, one thing we can do is, we can look at the signal from gut microbiome and then measure the response of those DNA elements. Or we can, in the animal, we can give them different dietary factor and see what's the response when the element carry, for example, different variant. All the other things, we can introduce the library into the animal at different age. And then we can start to understand during the aging process how the element responds. So I will quickly go through some ongoing work. We plan to submit in very soon. So we finished the screen, and then overall we identify 13% of elements we test here are activating elements, so which means they can activate transcription, and about 3% of them can replace transcription. And when we look at those elements using the data generated from human liver, we see the activating element, they are significantly enriched for H3K27 acetylation. And also, we also find that the target gene regulated by those elements in human liver, they also tend to have a higher expression level. And for the target gene controlled by the replacing element, they have a lower expression level. And those genes are important for example, immune response, or for mitochondrial translation, or response to the signal. So they are play an important role in human liver. And then from the data we generate, we also able to identify one functional rare genetic variant related to liver cancer. So here we use a cohort study generated from Singapore Chinese health study. So it's a long-term study. They collect the sample from the individual when they are 40, 50 years old, and then every year they ask those individuals come back and then give the DNA and blood sample. So after that, so they can identify there are some genetic variant they are associated with liver cancer, and then when we incorporate our data we generate here, we identify one of this variant. They are identified from our screening because in HepG2 you don't see the signal here. And then we also show when we introduce the element contains variant into mouse liver, they also have a stronger transcriptional activity. And then we also identify the target gene regulated by those elements is this gene nearby called INF2. And this INF2 has been shown based on the TCGA data. When the liver cancer patient has a higher expression of INF2, they tend to have poor survival over time. And the next thing we want to do is we want to know in our body, our genome, how many of them actually can sense the signal from gut microbiome. So here we repeat the study. We introduce the library into wild type or germ-free mice, and then by sequencing, we can tell and identify which element is down-regulated or upregulated under germ-free condition. And one thing very interesting from our data is we see a lot of regulated elements, actually their activities depend on the signal from gut microbiome. And then here, by performing a motif analysis, we identify a key motif called NRF2, which is important for oxidation, stress, Actually, here you can see a lot of elements when they carry this NRF2 motif, they tend to lose activity under germ-free condition. And then previously, recently there's another group from Vanderbilt. They also identified there's a gut microbe. They can secrete the metabolite to activate oxidation stress in the body. So from here we also want to do the validation, right? Because everything here is just by sequencing. So one thing we did here is we pick up some target gene we identify from our data, and here we do a very simple experiment. So here we want to know those target gene, their regulation is mediated by the signal from gut microbiome. So here we use HepG2 cell. We think it's like a germ-free condition because we capture them in the lab. There is no signal from bacteria. So here we can grow microbiome community and then isolate the metabolite, and then we give those metabolites to the hepatocyte cell. And here you can see, indeed, after the metabolite treatment, 3 of the target gene, their expression become significantly upregulated. And then one thing we do further is, very interestingly, if we change the composition of gut microbiome using the peptide, as I mentioned to you in another study. So here you can see after peptide treatment, you can see that the firmicutes, the abundance of firmicutes decrease, but when we provide the control peptide, it's more similar to the control. And then here, when you look at the gene expression response, and then here, when we give them the wild type, the metabolite isolate from wild type community, we can induce SEP4 expression. But here, when we use the metabolite isolate from the peptide treat community, it cannot induce the expression anymore. And this regulation is very gene specific. We also have other data to show it's not all the genes have the same response. And lastly, we want to also address what's the metabolite secret from the microbiome community contribute to gene regulation in the host. So recently, we developed a new delivery method. Because one challenge in the field is once we doing the sequencing experiment, we identify certain pathways are activated or downregulated in the bacteria. We don't have the genetic tool to deplete that gene or introduce the mutation. So because in E. coli we can do that quite easily, but in the wild type bacteria we still don't have this ability. Usually the typical approach we use is by conjugation. So here we find the cyclin peptide we used in the previous study, actually it can help to deliver different molecules into bacteria. For example, here we find after peptide treatment, now we can deliver a molecular beacon, which is a short DNA oligo into bacteria community. So you can see the fluorescence increase almost 95% after the peptide treatment. And we also can deliver Cas9 guide-RNA complex into the bacteria. So here by design the guide-RNA targeting to 16S ribosomal gene in the bacteria, we are able to deplete the specific bacteria in the culture. And then here just we are also trying to identify which species we can do the genetic engineering. So this is like a platform we developed. So we can by fluorescence sorting and grow them back on the culture, we are able to identify what's the sequence and what's the species we can deliver to. So for example, here just the example from 2 different peptides. You can see the peptide number 1, they are enriched for firmicutes and bacteroides, while peptide number 2 also enriched for actinobacteria and proteobacteria. So the last slide here is so here hopefully in this presentation I try to introduce the effect of gut microbiome. So now we have some new tool we can use to characterize the key microbe and metabolite, and also we develop in vivo system, we can understand how host genetics responds to different signals from the environment. And the next thing we plan to do is we want to look at the dietary component from Asian diet. Because we know different regions, they consume different diet. So in Singapore, there's we are working on put up a large grant together. We want to look at how Asian diet affect the gut microbiome and health in the region. So I just want to thank you all my collaborators and the students. And then also, in Singapore, we also have some graduate, some opportunity for graduate students. So if you are interested to pursue a PhD degree or even just an exchange program, 2-year program, you can stay in your local university for PhD for 2 years, and then you can come to Singapore to do research for another 2 years. We also provide this exchange program. And then also, we are a new group, so we are still looking for the postdoc candidates. Thank you.